Thanks for joining the session on acquisition and pre-processing methods. We have 6 nice talks lined up. Just a couple of announcements before we start. Please turn your phones to silence. And also try to avoid moving around too much during the talks. I think it's disrupting for the speakers and not very nice. We're starting with a bit of a delay. I think that was good to just let oxygen levels recover a little bit. But without further ado, I'd like to introduce Christoph Leuze, and he will talk about fiber orientation measurement using diffusion, MRI, and clarity on the same human brain tissue.
Yes, so thank you very much for the introduction. Fiber diffusion tractography currently is the predominant method to measure fiber tracts in vivo in the human brain. It is an indirect method, so we measure the water diffusion profile, and from that we reconstruct the fiber pathways. And because it's indirect, it's also prone to certain measurement errors. For example, we can have the problem that the fiber tracts are too small or they're overlapping with too many other fiber pathways, which could lead to false negatives. Then there's the other issue that we might have a fiber tract which jumps to neighboring unconnected fiber tract, leading to these false positive results. Since these issues remain, we need to find methods to improve the fiber tracking, and one way for that is to find a gold standard to compare diffusion MRI to. And so here we propose to use clarity as a 3D histological method to visualize fiber pathways and establish it as a comparison to diffusion MRI. So clarity is a direct method. We directly visualize these fiber pathways so it can show us the real underlying tissue structure. So clarity is a 3D histological technique compared to 2D histology, where we section a brain into thin slices and stain them for cellular molecular markers and then combine them together to a 3D volume. In clarity, we also stain for cellular molecular markers, but we do everything on a 3D cube of tissue. In order to do this, the clarity process involves several steps. The first step is the hydrogel embedding. So we have our uncleared tissue specimen, like you see here in the photograph. Then we embed it in a hydrogel. The hydrogel is an acrylamide hydrogel that binds to the proteins and the nucleic acids and sort of locks them into place in this hydrogel network. Once it's embedded in a hydrogel, we wash out the lipids and everything else that did not bind to the hydrogel. And after washing out the lipids, the sample becomes more transparent, and what we're left with is this hydrogel network with the proteins and nucleic acids still at their original positions. Once the sample is cleared, we then mark these proteins, nucleic acids, whatever we want to visualize, using fluorescent markers, and then finally image the whole thing using, for example, a light sheet microscope or a confocal microscope. Here's an example of clarity in a mouse brain. So stained for various cellular components. In reds, you see cell bodies. In green, neurofilaments. In blue, interneurons. So clarity was developed for mouse brain, works very well for mouse brain, but now here we are the organization of human brain mapping, and myself, probably many of you are very interested to see how that works in human tissue. And for that, there are first certain things to keep in mind. So there's a difference in clarity in human and mouse tissue. In humans, we generally have larger structures, which calls for larger specimens to clear. They're strongly myelinated, lots of lipids in the tissue, and they're drop fixed compared to perfusion fixed in mouse brain. All this leads to much longer clearing times in human tissue. Human tissue, we always have to stain. So in mouse, I didn't really specifically mention it. We can sometimes use transgenic animals that express fluorescence in a part of their neurons. In this case, we could skip the staining step. In humans, we always have to stain, which can also be quite time-consuming. In humans, generally, if you have larger samples with gyri and sulci, after clearing, they become softer, they are more difficult to handle, become more fragile compared to the little ball, like this little mouse brain. This is a bit more complicated. And also the microscopes, they are often suited well for small samples, for mouse brains. For larger samples, like for human samples, there might be some issues, such as, for example, limited light sheet penetration. And then, of course, if you have larger samples, we have more data, and more data is always more difficult to handle than less data. So now I come to the goal of this study, and this is to develop a comparison pipeline to compare human diffusion MRI and clarity results. And this involves first the tissue collection. Then we perform the structural and the diffusion MRI on the uncleared specimen. After the MRI, We then clear the tissue, perform the whole clarity process, clearing, staining, imaging. Once we have the clarity data and the MRI data, we need to find a way to align these two different types of data sets. And once we have them aligned, we need to find quantitative measurements, quantitative ways to compare our results. So the tissue collection, after collection of tissue, the structural and diffusion MRI, they were pretty standard that we did. I want to talk a bit more in detail about the clarity method. So this here shows a cleared specimen. As you can see, since you probably don't see much, it is really clear after clearing. It takes really long for the human brain, so a 2 millimeter sample, washing it at 37 degrees in detergent can take easily 2 months. After clearing, we stain it. For staining human tissue, there are mainly like two different methods. First is the small molecule stain, like you can see on the left. So the propydium iodide we see here stains, for example, DNA and RNA, works as a cell marker. And you can see, for example, these different differences in the cortex and cortical layers. The small molecule stain is very small, so it penetrates the tissue very well and leads to a quite quick and homogeneous stain of the sample. However, there is unfortunately no small molecule stain that works well to visualize large fiber structures. And in order to visualize fiber structures, therefore, we use immunostaining. So we use antibodies. In this case, on the right, you see the neurofilament stain. Antibodies are much larger than the small molecules, so they penetrate not as well into the tissue, and it needs longer time in order to penetrate all tissue and achieve this homogeneous stain. Once we have the MRI and the clarity data, we need to align them. So we were trying to figure out for a long time, okay, what are the best fiducial markers? Can we place microbeads in our sample that are both visible in structural MRI or clarity, or some glass rods or something similar? It always turned out that it's very hard to find something that survives the whole clearing process, that stays attached to the sample during the whole clearing process, or that does not destroy the sample. And then we found that actually the blood vessels, they are both very well visible in the structural MRI, and they're very well visible in the autofluorescence image of the clarity data. And we can also recognize the same blood vessels very well, like they preserve their shape in both the clarity and the structural data. And therefore, they can act as a sort of natural fiducial marker that allow us here on the right this affine registration between MRI and clarity data. And finally, if we want to extract certain features from the clarity data, we measure the structure tensor. So structure tensor is some sort of edge detection technique which measures intensity, signal intensity gradients at the boundary of fiber pathways, and this provides us with the overall orientation of our fiber structure. So now we used all this method and performed the whole pipeline on one sample. So here you see one tissue cuboid extracted from the thalamus of a human brain. We performed a high-res structural MRI scan and diffusion MRI scan, and then we cleared our sample. And now let's see whether the video works or whether you have to, yes. And now here you see the cleared sample stained for neurofilament in green, and in red you see the autofluorescence channel. So the autofluorescence shows very well, for example, the vessels inside our tissue. After clearing the tissue, so now we have the cleared brain, we have the MRI measurements, we align them using the blood vessels, so they're blood vessels that are visible in both image modalities. Aligning them, we use the structural MRI as an intermediate to then align the lower res diffusion MRI to the clarity data set. And after aligning them, we measured the structure tensor in the clarity data set in the same space as the diffusion MRI data set. And let's zoom in a little bit in order to see what structure tensor did here. So the structure tensor you can see here is very well aligned with the fiber structure of our clarity data. In order to compare how well the structure tensor and the diffusion tensor aligned, it's easier to look at the primary direction of both modalities. So here at the top in the clarity data, you see in red the primary direction of the diffusion of the structure tensor. In blue at the bottom edge, you see the primary direction of the diffusion tensor. And on the right, they are overlaid. If we now measure the angle between the red and blue, between the structure tensor and the diffusion tensor, we get the following color map. So these large blue regions show a very low angle between structure and diffusion tensor, so a very good agreement between structure and diffusion tensor. We see also at some regions, like here at the front of the sample at the right of the screen, we see some regions where there are some differences in these modalities. Right now, we are still at a stage there could be several parameters leading to this. So one thing could be we had a fine registration between the clarity and the MRI data. Could be that because the clearing leads to some distortions, that especially at the side of the tissue, we need some nonlinear registration. Could also be that just the simple model, the diffusion and structure tensor, are not good enough to appreciate certain crossing fibers, so that we need to apply some crossing fiber models in this region. And of course, there's always a possibility that, for example, the diffusion tensor, the diffusion weight imaging doesn't pick up the correct directions in this region. To sum up the results, so we proposed a pipeline that allows comparison of diffusion MRI data and clarity in the same specimen. Then I showed that the human clarity protocol, it needs some modifications from the original mouse protocol that had been described extensively in the literature. And as the next steps, we try to adapt the clarity protocol to work really consistently also for larger samples. And then, like I mentioned in the last slides, we also need to perform a comparison for more sophisticated diffusion MRI models that take into account crossing fibers. Yeah, thank you very much. I want to acknowledge our whole team that is also working on this project, and if you have any questions, please go ahead.
Indeed, we have time for 1 or 2 questions, if there are any on the floor.
Hello, Alan Mackenzie Graham from UCLA. Thank you for a fascinating talk. I was curious what wavelength you were measuring the autofluorescence at. And secondarily, you spoke very generally in terms of the amount of time for the antibody probing to take. Ballpark, how long does that take for a primary, secondary antibody combo?
So it depends. So to the first question, the autofluorescence is, I think, 448 nanometers. Then the neurofilaments is 600 something. I don't exactly know the exact number, 660 or something nanometers. For the time for the staining, it really depends on the size of the sample. So this 500 micrometer sample was roughly for the small molecule stain goes really fast, a couple of days. For the antibody stain, it can take easily a month. If you go to bigger samples, 2 milliliters or more, it can take several months. So then you have the clearing, which takes very long. You have the staining, which takes, again, equivalent. Yeah.
Thank you.
Hi, I wonder if you could just give us some, sorry, I'm Tim Behrens. I wonder if you could just give us some impressions of when I saw that clarity data, which is beautiful, by the way, I couldn't really figure out to what extent you could follow individual axons, particularly if there were crossing fibers, et cetera, I mean, and just wonder if you could give us some impressions of how it fares compared to PLI, for example.
Yes, so now in this case, for the neurofilament stain, you mainly saw fiber bundles. It was hard to follow individual axons. It would be nice, so if you look at the transgenic animals, for example, mouse, you see these individual axons. Right now, we don't have a suitable stain for the human tissue that allows this. We might try a little bit sparser neurofilament staining, but...
and so the advantage in doing this over PLI is mostly because it's a 3D technique rather than a 2D technique?
Yes. Yeah.
I see.
I have a quick question. You mentioned that it takes a long time for the clearing process, right?
Yes.
Would it be possible to scan the images on the MR after the clearing, just so that you can have a look at the distortions that you get?
So yeah, I actually tried that. I talked about it actually during the last OHBM. So what happens after clearing is that your tissue contrast completely vanishes, which is interesting. So you have still you have your hydrogel network, you still have your proteins, your cytoskeleton more or less in place, but your tissue contrast T1 diffusion completely vanishes. So it's hard to use that as a reference for the registration.
Okay, thank you.
Yeah.
Okay, let's thank the speaker again and move on. Our next speaker is Wietske van der Zwaag from the Spinoza Center for Neuroimaging in Amsterdam, and she'll be talking about distortion-matched T1 mapping.
It's very bright. Thank you. Good morning. So I'm going to start with a very obvious sentence in the introduction, but I think it's part of the talk. So as we've learned over the last decade or so, we've seen that 7T repeatedly can provide good enough bold contrast to noise to acquire fMRI data at sub-millimetre resolution. And that was a big step, because this sub-millimetre means that we can acquire several voxels across the depth of the cortex, and that means we can look at functional responses as a function of cortical depth. And not surprisingly, there's been a whole slew of papers presenting laminar fMRI. I just put some examples on the screen here, but there's many, many more. And one thing that these have in common is that all of these people have put a massive amount of effort into moving their anatomically defined layers onto their functional data, which is always distorted in some way slightly different to their anatomical data. And that problem is made worse by the fact that these have different contrasts. And the T1 images, the anatomical images are T1 weighted. We need that to have a good separation between the gray and white matter in the CSF, whereas the functional images have to be T2 star or T2 weighted to make sure that we have bold contrast and have some amount of efficiency to keep the TR under control. So one logical solution, we'd say, would be to use T1 weighted echoplanar images, which would have the same distortion but different contrasts. And this is anything but a new idea. Again, I put some of the references up. I did this what seemed now a lifetime ago almost, and even then it wasn't very new. But it has had some renewed interest in this past year, actually. There were several talks at ISMRM showing T1 weighted APIs. The problem with this generally is that it's terribly inefficient. So the purpose of this talk is to show you a sequence which is efficient and fast and provides T1 weighted images with matched distortions to the functional data. We've called this T1 with two 3D EPI readouts. So one of which you see in the gray block line out here. It's a standard 3D EPI as you would use it for a functional acquisition. And as you can tell from the name, we have two of these, and they're preceded by an inversion pulse. So the first image after the inversion pulse is very strongly T1 weighted, as you can see from the image at the top. The second one is so much later that there's barely any T1 weighting left. And these two images, otherwise they are the same, so they have the same T2 star weighting. They're still functional images to a great extent. All right. So this pair of images we can combine via the same algorithms we use for the MP2 range, so the anatomical images, and that combination effectively removes all of the T2 star weighting, most of the B1 effects, as well as the M0 contrast. And that would then leave you with a pure T1 weighted image. Right, so as you can see here from the bottom, and because they are purely T1 weighted, you can then also generate a T1 map from these. And the reason this is so efficient is that, if you just look at the sequence scheme, if you would be able to squeeze your entire fMRI acquisition into one of these gray blocks, then the time between two inversion pulses would be enough to acquire anatomical, and that long TR it's long, but it's about 10 seconds. So that would give you 10 seconds to acquire anatomical data. Obviously, that would be a bit wishful thinking. Usually you might want to either average to increase your SNR, or if you can't be fast enough, if your functional acquisition is too slow, then you'd have to chop this into various bits. So you could either acquire half a K space in each block or even a quarter. So what we're doing here is then compare this to the standard MP2 range. So it's 0.6 millimeter isotropic, 11-minute acquisition. It's not particularly fast. And the functional reference that we took is a 0.7 millimeter isotropic 3D EPI protocol from Utrecht, which has been used several times already to do laminar fMRI with. So it's not a carefully chosen example. It's just what's around, what works well for laminar fMRI. And then we morph the T1 to 3D EPI to fit this 0.7 mil functional data with either two blocks, so acquiring half a K space following each inversion pulse, leading to a total acquisition time of 80 seconds for 4 averages, or a slightly shorter block where you can choose the inversion times a bit closer to their optimal value, and that gets you to just over 2 minutes total acquisition time. So then the images. These are 4 subjects. The acquired with even different RF coils, but they're all visual cortices. And if you look at the left row of slices, then for those you see that that 2 block where we have a slightly longer first inversion pulse, it has a little bit of a washed out look to it, and that is because that inversion time is a bit longer and it doesn't quite get you the optimal contrast. The 4-block option looks very much contrast-wise like the MP2H, and remember it's only 40 seconds longer. Same thing for the T1-maps. Here, of course, the contrast is not different because the lookup tables are different, and then your contrast will work out to be the same, but the 2 blocks are a bit noisier than the 4 blocks. So again, you get a more solid estimate of your T1s by using a slightly shorter readout. So of course the important thing is then how well does this co-register? And there are many different ways you can go about co-registering. There are many distortion corrections you could apply. I tried to make it as simple as possible to get the comparison fair. So starting here from the MP2H, so this is the same slice. I took the MP2H through the visual cortex, just one of them, and then I overlaid the contours just to give you a visual marker. If you overlay those onto the mean EPI, which is what the MP2H was co-registered to, then you'd think, and this is a big room, so especially if you're sitting at the back, this looks pretty good, I imagine. However, if you zoom in, then you see that it's really not very good at all. You're having so there's these 3 blue arrows which indicate either the white gray matter surface, the gray matter CSF, or the gray matter to the extra brain surface. And for each of these 3, in places, it'll deviate for more than a voxel in the mean EPI. Right, so and obviously if you're trying to do anything laminar-wise or cortical depth related, if you're off by a voxel, that means you're off by 1/3 of your total cortical depth. That's completely unacceptable. Now, encouragingly, if you take the T1-weighted 3D EPIs, so these are the same slices, they're separately co-registered to the mean EPI, but I've overlaid also the MP2H contours, then you see that they have the same shifts as the mean EPI. Right, so obviously if you then overlay the activation that we get from the EPIs onto the T1-weighted EPIs, then you see that follows perfectly the grey matter strips. And if you, say in a better approach of doing this, you would start from the T1-weighted and then work up contours from that. So that's what I did here. This is a slightly different acquisition, now 0.8 millimeters, where we're doing a motor task. If you're overlaying the contours that I've now generated off of the T1-weighted 3D EPIs onto the mean EPI, then again, it looks globally pretty good. You can already see that there's some problems in the MP2H. If you zoom in, then it's even more obvious. They fit pretty much perfectly on the rightmost insert, where there's a very high fidelity. If you're looking at the bottom zoom in of the MP2H, then you see that there's massive problems again. So, again, as you would expect, you see then also that these motor responses follow very well on the grey matter in the T1-weighted 3D EPIs, both in the motor cortex and in, I was going to point this out, but the secondary somatosensory cortex, which is just a bit below, if you can find it. So one thing that I really wanted to do with this sequence would be to have a look at the cerebellum, because there we always have a lot of co-registration problems and small residual distortion issues. So I tried a bigger slab now at 1 millimeter isotropic resolution, and you can already see even in just the images that there's a problem with a fat sat here, so I should have probably spent a bit more time on it. However, the co-registration still works very well. So if I zoom in, then you see that the T1-weighted 3D EPIs, which are the ones that I've drawn the surfaces off here, overlaid onto the mean EPI, they fit very, very well, whereas there are small shifts between the grey-white matter boundaries of the MP2H and the T1 to 3D EPI contours. What you can also see is that this 1 millimeter isn't quite high enough spatial resolution to really define all the fine structure of the cerebellum, so we should probably redo this with much higher spatial resolution. So one thing that I would really like to happen for these acquisitions is to have a fully automatic pipeline, and sadly all of those that are around are based on whole brain data. So also set up a whole brain protocol, which you could also match the distortions. It would just then take a bit longer. So this is now 14 case-based segments per inversion and only 2 averages. So in 2.5 minutes, you still have a T1-weighted, now whole brain API image. And the segmentation with just a brute force SBM works extremely well. So I think this is also a good sign in that if you put it through a free server pipeline, like in the Renval paper from this year, then that would also work. So I think I've used up all my time. So let's get to come to the conclusions. The T1-weighted images that we acquire with this MP2-RAGE-like sequence can be acquired in just about 2 minutes. And we can also use the whole brain version of this to segment and probably define layers with standard software, meaning that we could just skip our anatomical acquisitions altogether, and that would give us a lot of time to then improve on these as well. For the automatic pipelines, it would be terribly helpful if we could do the segmentation of partial brains automatically. However, an alternative would be to acquire distorted whole brain T1 to 3D EPIs, or else to use a nonlinear warping of the anatomical to the partial brains, which, given the BBR algorithms that are around, should work pretty well. So with that, I come to end. I'd like to thank my co-authors for being so very helpful with this project, and you for your attention.
Okay, thanks, Wietske. Nice talk. Needless to say. So any questions? Let's take one and then move on. I actually have a lot, but I'll ask them offline. Okay, let's move on to the third talk. Let's thank Wietske again. And the next one, that's work by Laurentius Huber from the NIH Functional Neuroimaging Group. This talk will be given by Daniel Handwerker, and it's on effective conductivity measured with layer-dependent resting state blood volume fMRI in humans.
Okay. So for those of you who know Laurentius, he is unable to attend. He's done great work on this project, and it's my pleasure to be able to present for him here. So to start out, we have a brain, and we know that a lot of there's different functionality in the cortex, and most of the brain is divided up into 6 cortical layers in the cortex. For this specific study, we are looking at specifically an area where this has been mapped pretty well in mice and other animals, essentially M1 layer connections. So if we were going to use fMRI to look at layer dependence, we know that postsynaptic activity is going to be a major part of that signal contribution, and that would be in the form of inputs into an area. So if we're looking at M1, we're getting inputs from the other places in the same M1, contralateral M1s, S1, thalamus, and other things. And you can see that those inputs come mostly to the upper layers, like around layer 2, and the lower layers around layer 5. But, actually, just to go back. And so if you're having some sort of task that is having all those inputs coming in, if you wanted to hypothesize what the fMRI response should look like across layers, you would expect to see 2 peaks across the layers, around those upper layers and those lower layers. If you're looking specifically at inputs coming in from S1, you would expect to see only 1 peak. So that's the expectation and the hypothesis we're testing in this study. Of course, to study layers, you need really high resolution. So we're collecting data at 0.75 millimeter resolution here. Of course, resolution isn't everything. So if we take this M1 cortex and we look at the bold signal at 0.75 millimeter resolution, it's still kind of a large blurry area because, as many people here know, bold is an oxygen-focused contrast, which means that the signal is going to be still dominated by larger veins. So it's going to blur the signal towards the surface vessels. On the other hand, if you can image volume changes are going to be dominated by the areas that relatively change the most, which are the capillaries. So you have the potential in volume to get a much finer resolution signal in those finer capillaries and distinguish layer-specific effects. So we are doing 2 different tasks to probe if we can see this difference. The first task is resting state, so it's a 12-minute resting state scan. And the second task is finger movement. So it's a 12-minute task with the task is 30 seconds on, 30 seconds off, block design, with 2 conditions. The first condition is finger movement with finger tapping. So the volunteers are moving their fingers just very simply, repeatedly, and touching their fingers together every time. So in this case, we're expecting, obviously, all motor inputs are going to be active. In addition, you're going to get a fairly large sensory component from the fingers touching together. The second condition is finger movement without finger tapping. So you're doing the exact same motion, but your fingers aren't touching. So everything that was involved in the first task should still be there, but that added sensory component of the touch should be reduced or not be there at all. To collect this data, we are using vascular space occupancy imaging, or VASO. So VASO is a multi-T1, a two inversion pulse sequence where first you're doing a global inversion to null the blood signal. So you can see that on the red line at the bottom, where the blood signal is nulled. And then after a delay time, you're collecting the first readout, which is going to have the blood signal nulled there. And then as the blood signal comes back, you're collecting a second readout where you can see the blood signal. And essentially the ratio of those two images is your VASO image. So it's the relative change. So it's optimized to see how much volume changes across those two images. Other parameters, this is 3D EPI. I'll self-promote a bit and say that if you come by my poster tomorrow, there's a little sidebar on why we're using 3D EPI for these as opposed to simultaneous multi-slice for acceleration. Other parameters, it's flash scrapper 2. It's a TR of 3 seconds, so each of those two volumes is collected 1.5 seconds apart. So it's 1.5 second tag, then another half second control, but to get both of them, it's a 3 second period. The in-plane resolution is 0.75 millimeters. The slice thickness is 1.8, so it's bigger. We need the signal to noise, but what we do to optimize that is we're doing a very focal field of view. So it's really focused on this S1-M1 area. And Laurentius align the slices so that in individual subjects, so that the in-plane direction is optimized for this area that we're measuring specifically. So the in-plane direction that really gets the layers of S1 and M1 is 0.75 millimeters to maximize that layer-specific resolution. Other experimental setup, this study was done across 10 volunteers and 18 experiments. It's a 7T, 32-channel coil. There's a lot of stuff on optimizing tSNR because we're pushing signals to limit to get these really small voxels. I'm not going to cover that here. I can talk about this in questions or afterwards if anyone has questions, or you can contact Laurentius. And some of the optimizations have been based on other people's work too. So layering, I can go through the slides really quickly because we just heard an awesome talk that essentially was the same idea, which is because we have these two inversion pulses, we essentially, as part of collecting VASO data, get a free quantitative T1-map from the result, which gives us a really nice border of gray matter and CSF. And if you take the epiphase information, we actually get a very nice gray-white boundary. So we have the two boundaries of the cortex in the same distortion-corrected plane as the data we're caring about. And what we can then do is take those two borders and line up with equi-area distributed layers in between. So we did 20 layers in between here, just to get sort of 20 thin strips of cortex to compare layer changes with. The next step we did was we smoothed within layers. So this is how the data looks to also give us this tSNR boost. This is how the data looks without smoothing. And you can see as you increase smoothing filters within layers, you're getting a much more defined layer effect. And you're already seeing in VASO, we're seeing sort of a separation between layers while bold is still blurred because it's not distinguishing the layers as much. And here is the images sort of as a static image. And please stay around for the session for another great talk by Anna in 20 minutes, which is focusing on this topic specifically. So we're analyzing data with two different methods. The first method is a seed-based analysis, where we have a seed in S1, and we're looking at correlations to time series in all those 20 20 subdivided layers in M1. And we get these depth profiles from the orange, which is near CSF, in the upper layers. Orange is in the upper layers, and blue is closer to white matter in the lower layers. Similarly, and we also use an M1 seed in a different slice. And I'll talk about what those images are. Those are the key results, but I'll show them more on the next slide. For the intracolumnar analysis, we're essentially drawing just little columns across the whole cortex. We're taking the mean within those 20-time series in the cortex, in that column, and calculating the correlation to those means. So it's sort of a measure of common variance across columns. So for that connectivity, now this is the figure from the last slide that's zoomed up a bit bigger. What you can see is when the seed is S1, there is only an upper peak in those higher layers. But when the seed is M1, we're getting 2 peaks in the upper and lower layers. So you can see this flipping back and forth in M1 seed, S1 seed, where when the seed is in M1, we're getting 2 layers in the green circled seed region, and in S1, we're only getting 1 layer. And you can also see the seeds flipping back and forth underneath. And again, looking at that model underneath, you can see that this fits with the hypothesis of what we expected to see, with just S1 contributing to those upper layers. So we've also validated this with the task data. So this blue line here is the correlation magnitude with the finger movement with touch. And again, because there's the touch, so you have a signal in upper and lower layers, around layer 5 and layer 2. But if you remove touch, you're getting the same response, but that upper layer response, which has the extra touch input, is reduced. So you can see going back and forth with touch and without touch, you're getting a reduction in that upper layer when the touch is removed. And we can just simply subtract those two, and you see that there is a differential response in those upper layers when the touch component is removed. Interestingly, I began this talk saying that this is not possible with bold, but if you look at the with touch component on the top and the subtraction on the bottom, we actually do see this effect in bold. The trouble is that you can see the biggest signal in bold is in the CSF, where those draining veins are. So if you didn't know exactly what you were looking for, it would be washed out and be really hard to identify this without seeing the VASER data too. Finally, this brings us to the potential of how we can build up, speculate about what's possible with this method going forward. So talking about that variance mapping, we took, for every column, we made a profile of where the most variance is, and we used hierarchical clustering across a slice to see how it clustered. And it actually clustered quite well. So in lower areas, in parietal areas, we saw things clustering in higher levels, near white matter. Then we saw some areas with a double peak, some areas with upper dominance, and some areas that it's hard to visually classify. And this kind of fits into sort of a feed-forward model of what different regions in the brain might be doing by going back to conceptual studies like by Feldman and Van Essen. So it's a potential this might be. There's issues. There's still work to be done on this, but there's a potential this could be a future type of analysis too. And finally, in addition to the co-authors that were shown at the beginning, there's a lot of people who have contributed to this project in various forms, and we'd like to thank you.
Thanks, Daniel. And any questions? We'll take one or two.
Nice talk. Thanks, Daniel. Your seeds in M1 and S1, in which layers did you place them? So or are they across layers?
So the seeds are across layers.
The seeds are across layers.
Yes.
And from a motor perspective, it seems like so you're looking at least resting state connectivity. I mean, there's back and forth communication between S1 and M1, and there's also plenty of activity when you just move your hand without touch in S1. So there's a efference copy going in S1. Can you speculate why? I mean, why I wouldn't have from a motor neuroscientific perspective, I wouldn't have actually expected the results to turn out so clearly as you showed. Can you speculate why you think that those other connections wouldn't have played a role here? The forward connections from M1 to S1 specifically.
So from M1 to S1?
Yes.
So the depth profiles we're showing are just in the M1. So I'm not showing sort of the layer dependent responses to S1. There are actually some challenges with that because just from a physics point of view, S1 is thinner cortex. So to get the same resolution of cortical layers in S1, we actually would need even smaller voxels. But in general, I've been trying to be very careful here talking about relative changes. So removing the, finger tapping versus not finger tapping doesn't mean that, without touching the fingers does not mean you're getting no sensory input. It just means it's reduced. So we expect that there could still be something there, but it's the reduction that I think was the key effect, not that it was non-existent versus existent.
Okay, one more question, then I'm afraid we should move on.
Hi. The first picture you show with the columns, was that an S1 or an M1?
You mean so dividing the whole thing into the whole brain into columns?
Yes.
So that analysis was done whole brain. I should say that was actually sort of a different preliminary data set where we got whole sliced activation with 0.8 millimeter voxels.
And like, how do you define these columns? Like, what's the criterion to define like when one column ends and the other begins? So
I do not know precisely what Laurentius did, but essentially, so you have the cortical ribbon, and each column has approximately an equal width. So I don't know quite how we define width around curving areas, but it's roughly every column is an equal width across the data going around the whole brain.
Thank you.
Okay. Let's thank the speaker one more time. And moving on to the next talk, that's given by Justine Beaujoin from Paris, and she'll be talking about post-mortem mapping for the inner connectivity of the human hippocampus using a diffusion MRI at 11.7 Tesla.
Thanks for this introduction. Okay. So, as you know, the human hippocampus plays a key role in memory management, and its connectivity is very complex because it's an information crossroad. But some of its pathways are already histologically known, like the polysynaptic pathway. And in this work, we investigated its inner connectivity using diffusion MRI, as well as its microstructure using diffusion MRI microscopy. But to reach such resolutions, clinical systems are not sufficient. So that's why we use the preclinical MRI system with a very high static field, which enables to reach higher spatial resolution. For instance, here you can see the difference between a human hippocampus acquired at 3, 7, and 11.7 Tesla. And there were also very strong gradients, so that enables to have a higher angular resolution and consequently a more accurate tractography. So we wanted to have anatomical and diffusion-weighted images. So diffusion-weighted images were used for studying tractography and microstructure and should be acquired with a very high B-value and still have enough signal. But the more you higher the B-value, the less signal you have. That's why the idea here was to infer the maximal usable value for the echo time and the B. So you can see the T2 and the mean diffusivity distribution acquired with a multi-slice, multi-echo, and a diffusion-weighted sequence. And you can see the two peaks for the T2, one for the white matter and one for the gray. So we use the second one for the computation because it's the highest one, and the hippocampus is mainly made of gray matter. And you also have the mean diffusivity value. So setting a maximum threshold for the signal loss, we inferred a maximal echo time of 60 milliseconds and a maximal B-value of 10,000 seconds per millimeter square. So in the frame of the HBP project, Dr. Mark Saxa from ULISH provided us a human post-mortem temporal lobe, which was scanned with a 11.7 Tesla Bruker MRI system. The acquisition protocol included a T2-weighted anatomical sequence depicting the anatomy at 200 micrometer. So here you can see the images. And also a multi-shell PGSE sequence with 3 different B-values and 125 direction datasets used both for tractography and microstructure study. So here you have an example of an image acquired at a B-value of 4,500 seconds per meter square. The first step was to compute the ODF field using the Hardy datasets. So here you have an example of the ODF field computed with an order of 8. You have the parameters used, and the pre- and the post-processing were done using the Connectomist toolbox. Then we manually segmented the hippocampus, the inner and the surrounding structures using the anatomical images acquired with a spatial resolution of 200 micrometer. And the choice of the segmented structures was done in order to observe the polysynaptic pathway. Most of the anatomical landmarks were taken from the literature, for instance, the way we segregate CA1 from the subicular area. And we also used the ODF fields computed before to ensure the consistency of the segmentation. And also, you can see here the orientation drift between ODF contained in the alveolus and the fMRI. So we segregated the alveolus and the fMRI using that. So we have the different step of the segmentation, and here the final result. So then we computed the tritogram using the parameters listed here, and from the tritogram and the obtained mask of the segmented structures, we inferred the polysynaptic pathway and the connectivity matrix of the segmented regions. So here you have the reconstructed polysynaptic pathway with bundles from the internal cortex to the hilum through the subicular area, then reaching CA3, CA1, subicular area again, and the alveolus and the fMRI. So this is our first result. Diffusion MRI enables to reconstruct inner pathways in hippocampus. But there are also very interesting results. If you look at the connectivity matrix, you have the connectivity in the head, the body, and the tail. And you can see that the connectivity is much higher in the head. And then we can wonder, what about the microstructure? If the connectivity is higher in the head, is there also a higher level of neurite density? And that's what we investigated using the NODI model introduced by Zheng in 2012. And we adapted it for an ex vivo sample. So no need to use a b-value of 200 seconds per millimeter square because we had no CSF to fit. And we also adapted the different parameters. And here are our results. So you have the intracellular volume fraction map. And it shows a higher neurite density in the head. To go further, we computed the main intracellular volume fraction for each gray matter region segmented before. And you can also see the positive gradients between the tail and the head. So we want to conclude that maybe, yes, there is a higher neurite density in the head, which is consistent with the results we had with the connectivity, showing a higher connectivity. And there is a last result we can comment. It's that the intracellular volume fraction map reveals the laminar structure of the cornea ammonis, showing 3 distinct layers. And if you look at the drawing from the juvenile book, you can see that layer 2 is made of soma. And on the intracellular volume fraction map, it appears very dark, which makes sense because it means a poor neurite density. And you can also see that it's much more easy to distinguish the different layers with this map compared to a classical anatomical scan on the left. So we have 2 main results here. First, the intracellular volume fraction map shows contrast consistent with the histology, and then it shows new contrasts, which enables to better distinguish the different layers. And to conclude, so we have seen that different MRI and diffusion MRI microscopy allow to probe the hippocampus at a mesoscopic scale and reveal its inner connectivity and microstructure. And regarding the histological validation, our sample is now being analyzed in Jülich by Dr. Mark Saxer, so we should be able to compare the results we had with its results and maybe have more information. So thanks for your attention, and I will just finish by acknowledging all my colleagues in Neurospin and from the HBP. Thank you.
Thank you for the very nice talk. Are there any questions from the floor? Nothing? I'll just have a practical one. I mean, regarding the acquisition. I mean, your system is pretty amazing in terms of the specs compared to the sort of human whole body systems that most of us work on. Yeah. How hard can you really drive them in practice? I mean, in terms of gradient slew rates and gradient amplitudes. I mean, your acquisitions are quite long considering those specs. So are you limited by heating or practical problems?
No, if I had problems with, I just didn't really understand your question.
Okay. whether gradient heating, eddy currents, and such like are actually real problems in those animal systems. Well, the specs are normally very great, but I'm not sure you can redrive them as hard as they look on paper. Never mind. In the interest of time, let's just move on. I can ask you afterwards. Yeah. It's not so interesting, maybe, anyway, for most people. Okay, well, let's thank the speaker again and move on. Okay, so moving on to number 5. This will be given by Anna, working with John Polimeni in Boston, and she'll be talking about improved tSNR of high-resolution fMRI with surface-based cortical ribbon smoothing.
Okay, thank you very much. Okay, so conventional fMRI studies, such as cognitive science studies addressing certain populations, usually expect activation size in range of multiple millimeters. And so the sufficient data resolution for these studies would be in range of 3 millimeters isotropic. High-field MR systems, however, such as FreeTest and higher, can provide much higher resolution than that, and so the question arises if using resolution higher than needed based on the expected activation size could provide certain benefits. So here we analyzed high-resolution 7 Tesla data smoothed along cortical surfaces, and we look at tSNR, temporal signal-to-noise ratio, to measure sensitivity. Our hypothesis was that anatomically informed smoothing can allow avoiding CSF and therefore noise, and so it can provide high tSNR in small voxels. So there are 2 different types of noise which we need to consider. Thermal noise, which is wide, spatially, temporal, and correlated, and it's characteristic for the high-resolution data, like this image here is 1.1 millimeter. And we can see at this image as well that the thermal noise doesn't really change with anatomy or tissue type. It will depend on the distance from the coil. The more problematic type of noise is physiological noise, which comes from head motion, cardiac respiratory motion, and it's spatially and temporally correlated. It's characteristic for the lower resolution data, like this 3 millimeter image here. And we can see that it varies with the anatomy and is also tissue specific. The highest tSNR in this image is in white matter, where the noise is the lowest, and the lowest tSNR will be in CSF, where the noise is the highest. It has also been shown that smoothing can cancel thermal noise, as it's spatial and correlated, and therefore can increase tSNR in high-resolution data. Most of the conventional studies still use volume-based smoothing, which is problematic because it can average across voxels which belong to different tissue types. So it can include noisy CSF or white matter with not much of the bolt signal. And we can see it here. If we apply the kernel, which is pretty standard size kernel, 5 millimeter, we can include some gray matter, but also CSF, potentially white matter if we move it around. And we can also include voxels which will lay far apart in the cortex, which will belong to potentially different functional areas. So to solve this problem, surface-based smoothing methods have been proposed, and they have been shown by multiple studies to increase sensitivity and specificity. So coming back to these large and small voxels, if this is our cortex, we have CSF at the top and white matter at the bottom. If we scan with 3 millimeter resolution, we'll have, let's say, the voxel grid like this. However, if we increase the resolution, each of these voxels will be divided into a bunch of the small voxels. So what is important here is that some of these large voxels will be contained in the cortex only, but some of them will have some CSF in them, and so the tSNR in these voxels will be decreased. So what we can do, switching to high resolution and using the small voxels, we can redistribute the voxels the way that we will avoid CSF. In addition, if we are using low resolution, the contribution of the small voxels within the large voxel cannot be really controlled. They will be weighted somehow in the same way. If we redistribute these voxels, we use high resolution, we can control the weights, and we can basically decide on the contribution of the signal from small voxels based on their location. So the question we are trying to answer here is basically what will be the smallest amount of anatomically informed smoothing, which effectively is using this redistributed small voxels to achieve tSNR higher than tSNR of large voxels. So the previous surface-based smoothing methods were usually just using one surface. Here with the small voxels, we can extend this approach and we can use family of surfaces across cortical depths, and we can apply the steerable kernels tracking the fold of the cortex. So this is our cortex again, the PL surface at the top. we can perform laminar smoothing along the surface, and the kernel, as I said, will follow the fold of the cortex. So basically, we'll look at certain depth, and we'll consider vertex and its neighbors, and we can do it on different depths. On the other hand, we can perform columnar smoothing, basically across the cortical depth. So we'll be looking at the vertex and the corresponding vertices on the other depths. We can combine these two approaches and perform a simultaneous laminar and columnar smoothing with this Weinkorg filter. Another combination of these two will be something like this diode filter, but we don't really analyze it here. So we scanned 5 healthy volunteers in 7 Tesla scanners and acquired data in 4 different resolutions ranging from 1.1 to 3 millimeters. What is important here that, as we can see, the parameters here don't vary only in terms of voxel size. They pretty much optimize on the way to match the parameters used in practice. But we also optimize them so this SNR of the 3 millimeter data is about 5 times higher than SNR of 1.1 millimeter data we tested on Phantom. So that's pretty much the factor we would have if only the voxel size was different. We acquired structural data with 0.75 resolution. We processed them with FreeSurfer to create 11 surfaces across cortical depths. And functional data were also processed in a pretty standard way and projected to surfaces. So to test our hypothesis that the appropriate smoothing of the small voxels can increase tSNR in the small voxels, we tested different smoothing approaches mentioned before. So we performed laminar smoothing with radius ranging from 1 to 6 vertices. Average vertex spacing was about 0.66 millimeters in our data, and we used weights 1 by distance. We performed columnar smoothing across all depths, and Weinkorg smoothing mentioned before, combining these two. We calculated tSNR on each cortical depth, and we normalized these values using mid-depth tSNR of 3 millimeter resolution non-smooth data for the comparison. So we first look at the tSNR across cortical depths. This is 1.1 millimeter data, and we can see, looking at the blue line just corresponding to the data with no smoothing, that is dominated by thermal noise. tSNR is pretty much constant across the cortical depths. If we look at the smooth data, the red and yellow lines, we can see that thermal noise decreases, physiological noise increases, and tSNR closer to the CSF is lower. And this is pretty much how it looks like in 3 millimeter data, either without smoothing or with smoothing. Again, we can see that tSNR closer to the PL surface is lower. So the results of laminar smoothing showed that 1.1 millimeter data, if we smooth them along the mid-depth, that's the solid blue line at the plot, with the radius of 2.8 millimeter, they pretty much achieve the same tSNR as, or higher tSNR than in 3 millimeter non-smooth data. On the other hand, if we look at the dashed line, which is corresponding to smoothing along PL surface, we can see that the radius here would need to be much higher as the slope decreases. And that's because of including CSF and physiological noise, therefore, and that basically decreases tSNR. For the columnar smoothing, as we can see here, that's pretty much what we would expect. For 1.1 millimeter data, we reached tSNR of about 50% of the unsmoothed 3 millimeter data. For 2 millimeter data, the tSNR was even higher than 3 millimeter unsmoothed data. And finally, the combination of these two. So with the Weinkorg filter of smoothing 1.1 millimeter data, we were able to decrease the radius of laminar smoothing to 1.8 millimeter to get the tSNR higher than 3 millimeter data, as we can see here. And that's compared to the previous 2.8 for only laminar smoothing. So what is interesting about this Weinkorg filter is basically that it allows the maximum radial smoothing, so basically include as much as we can, not leaving the cortex. And on top of that, we're adding the laminar smoothing with the minimum radius to achieve the same tSNR as the low resolution data. So this is just the illustration of all the smoothing methods. This image here is basically the original unsmoothed image projected to the inflated brain surface. This is the result of the laminar smoothing matching the tSNR of the 3 millimeter data, the columnar smoothing reaching 50% of the 3 millimeter data, and the final result of the Weinkorg smoothing. And what's interesting, you can see that the Weinkorg and laminar smoothing, they basically reach the same tSNR. But in the Weinkorg smoothing, you can still see that there's more fine details preserved from the original image. So in conclusion, we showed that 2D laminar smoothing of 1 millimeter data along mid-depth with radius 2.8 millimeters provided tSNR higher than tSNR of 3 millimeter data, which were unsmoothed. The columnar smoothing reached tSNR of 50% of the unsmoothed 3 millimeter data. And finally, the wine cork 3D filter allowed us to reduce the radius to 1.8 millimeter for the laminar smoothing. So basically, we proved the hypothesis stated at the beginning that anatomically informed smoothing allows us to avoid CSF and therefore noise, and so it can provide high tSNR in small voxels. What is important to remember here is that these numbers here are basically valid for our protocols. If you use different protocols, you probably need to optimize and find the right numbers to use. And this study is focusing on the sensitivity analysis using tSNR, but we also want to extend it to perform specificity analysis and look at the percentage signal of the change in cases of activation. Basically, investigate how much the large voxels will be contaminated by white matter. We also want to perform columnar smoothing, which will be weighted by partial volume effect. And I also want to note, so this work was mostly addressed to conventional fMRI studies, but high-resolution studies are already using small voxels and looking at small structures could also benefit from these different types of smoothing, because using the laminar or columnar smoothing could basically somehow anatomically perform like sampling on the anatomically informed way, which will allow preserving the spatial structure of the activation and increase tSNR in the same time. The final good news is that it was developed as a part of FreeSurfer, so it will be probably available soon. And just wanted to say that we have a poster on that, so if someone wants to have a longer chat, I'll be there tonight at the reception. And there's also another relevant poster, 1697, which you might want to have a look at. Thank you. And that's the thing.
Thank you very much for your talk. Do we have any questions on this? Everyone's baffled? Okay. In this case, let's move on. Thank Anna one more time and move on to the final presentation.
Thank you.
So for the last talk today, this will be given by Jeroen Mollink from Oxford and Nijmegen, and he'll talk about the fiber orientation dispersion in the corpus callosum.
Thank you. Thanks to the organization for having me here today. I would like to start with a general motivation for the kind of study I'm going to present to you in the next couple of minutes. So the general aim of my work is to merge microscopy with MR techniques to get a better understanding of the diffusion signal. So if we look at this figure, quite often we move from left to right, where we do an experiment in vivo and we try to validate our findings, for example, those derived from microstructure models, with post-mortem imaging and microscopy. But what I find particularly interesting is that can also move from right to left, where microscopy can be useful to reveal tissue features that normally wouldn't be noticed, but these may be strong enough to detect with in vivo MR applications. So in diffusion MRI, we're aiming to resolve fiber configurations within a voxel, and in recent years much effort was spent on resolving crossing fibers. Here we're focusing on more subtle fiber configurations such as fiber orientation dispersion. And quantifying dispersion may have the potential to improve current tractography paradigms, serve as a marker of local coherence, and as we saw at ISMRM this year, many white matter voxels are explained by a model that incorporates dispersion. So what we do here, we test our model in the corpus callosum, which is a single fiber population where accents are often assumed to be in parallel. So what we did, we scanned 3 ex vivo corpus callosum samples on a 9.4 Tesla scanner using a diffusion-weighted spin echo sequence. The voxel size was 0.4 millimeter isotropic. We used 2 different B values, each having 120 gradient directions per shell. After MR scanning, the samples were split coronally and processed separately. To obtain a reference measurement for the microscopic fiber orientation, we used polarized light imaging. And the other part was stained for myelin and astroglial fibers to study additional sources of dispersion in the corpus callosum. So let me briefly explain what polarized light imaging is. PLI uses polarized lights to quantify birefringence in the myelin sheet in thin brain sections. And this allows you to extract a variety of parameters from which fiber orientation is the primary target. So what we do, we acquire a set of raw images by varying the orientation of the polarizer in a microscope. And as a result, you can see this varying light intensity in the white matter due to the birefringence. And if we look at a single pixel, we can observe a sinusoid. And the face of this sinusoid then corresponds to your in-plane orientation. So using histology, we can highlight specific features of interest. Here we stain for myelin and astroglial fibers. While PLI provides us with a fiber orientation directly, the histological images require some processing. So what we do here is a Fourier filtering approach. So basically, if we have an input image, we transform it to the Fourier domain. And after some processing in that Fourier domain, we can extract the angular frequencies which correspond to the fiber orientation distribution, or the FOD. So this technique was applied to each histological image for a local neighborhood to obtain an FOD. And then from each of these FODs, we can extract dispersion, which is roughly set related to the width of this peak. So the MR model we're using is a two-compartment model. The isotopic part is modeled as free diffusion in all directions. And then we have an anisotropic fraction which consists of a collection of sticks which are distributed according to a fiber orientation distribution function. And in this case, the fiber orientation distribution function is parameterized using a Bingham distribution that allows us to quantify anisotropic dispersion. So the Bingham distribution we see here defines dispersion along two sets of axes. So dispersion parameters kappa 1 and kappa 2 then give the amount of dispersion. And from each of these values, we can compute an orientation dispersion index, which is 0 in case of anisotropy, so a stick. And if the fibers are isotopically dispersed, the OD is 1. So we apply the Bingham model to the MR data as well to the microscopy data. But the microscopy data only provide us 2D orientations, making kappa 1 meaningless in this case. So we use kappa 2 to quantify the orientation dispersion index. Then for MRI, we do the same. We quantify the orientation dispersion index based on kappa 2 parameter. So we do that for all the modalities and samples and then compare these against each other. So what I'm showing you here are the fiber orientation maps for the different samples across modalities. The in-plane orientation is color-coded according to the HSV color sphere or color space. And macroscopically, you can see some good resemblance between the used techniques. For example, here we see this striping pattern in the corpus callosum. This is then an overview of the orientation dispersion maps for the different samples and the different techniques that we've used. So the result after fitting the Bingham distribution to our FODs. Macroscopically, you can see that there is some resemblance between the used techniques. For example, we see high dispersion in the crossing fiber regions like the centrum semioval, and low dispersion in general in the corpus callosum. Although it should also be noted that, of course, we see some differences in some regions, and also the absolute values differ when comparing the color bar scales at the bottom. But for now, let's only focus on the corpus callosum, because that's what we were after. And especially here, we see an increased amount of dispersion in the corpus callosum. So in particular, a loss of coherence along the midline of this structure. So let us then have a closer look at dispersion in the corpus callosum. What I depicted here are the average dispersion profiles across samples for the different modalities. And as we saw in the previous slide, we see this increased amount of dispersion in the center of the corpus callosum. And if we compare the histology with MRI, we see that histology provides us with much higher, or little higher dispersion than what we see in MRI. And this may be explained by the fact that we, in histology, we have very high resolution data. So we really see these undulations in the membrane sheets at short length scales. And it could be with the current diffusion times we're employing in diffusion MRI, we're simply insensitive to these length scales. The fact that PLI shows you a bit lower dispersion could be explained that we're only sensitive to myelin, while other structures may contribute to the dispersion. However, if we do a correlation of these different profiles, we obtain very high correlation coefficients. So today I've shown you that we've been able to quantitatively compare dispersion across a variety of methods in the corpus callosum. By comparing these dispersion profiles, we find very high correlation coefficients. Those should be said that in terms of absolute values, we see some differences. The model we've been using in MRI assumes a stick-like fiber response function for an axon. And that may not be the most appropriate way to model a hollow axon. And finally, we want to know what's the biological meaning of dispersion in the corpus callosum. So to answer that question, we recently started a new project where we got access to the data from the Biobank UK project. And so far, we've applied a nutty model to almost 5,000 subjects. And we see similar trends of increased dispersion in the corpus callosum. We also have resting state data from these subjects, so we're now aiming to see if this increased amount of dispersion in the corpus callosum has a relationship to interhemispheric connectivity. But that's still work in progress, so hopefully I can give you an update anytime soon on that. So with that, I would like to thank you for your attention and thank my colleagues from these different groups.
Thanks for this talk with very nice images. Are there any questions? No question?
No questions.
So do you have any possible answer, speculative answer to your final question as to what the biological meaning might be?
Well, the hypothesis is a little bit that if you have increased dispersion, could it mean that if you have two homotopic regions like M1 and M1 on both hemispheres, if it's also projecting to other regions or to more other regions, would that mean if you have that you could have increased amount of dispersion because the fibers have to go to another region and therefore spread out a little bit more? But yeah, I'm still working on that, so I cannot give any conclusions whether that's true or not. So.
Okay, good luck with that work. I think you're all waiting for the answer.
Yeah, thank you.
So let's thank the speaker. And also thank the speakers, all the speakers from the session, which is now closed. So enjoy your lunch and the rest of the conference. Thank you.